Welcome to our Exhibitor Webinar Series, sponsored by MilliporeSigma. I do have a little note here for you, just to let you know the views and opinions expressed during this webinar are those of the presenters and do not necessarily reflect the official policy, position, or views of CASSS. And today's topic is navigating regulatory complexities along the cell and gene therapy life cycle from raw materials to release. And I'll bring on our friend in just a moment to do the speaker introductions. Oops. There we go. So go ahead, I'll bring on Mackenzie Landgraf from MilliporeSigma to do our speaker introductions. Thank you, Mackenzie.
Perfect. Thank you so much, Anne. Really appreciate the opportunity here. Hello and welcome, everyone. My name is Mackenzie Landgraf, and I will serve as your moderator today. As Anne mentioned, our webinar topic is navigating regulatory complexities along the cell and gene therapy lifecycle from raw materials to release. And I am here today with two of our expert speakers, Manjula Aysola and Dr. Alison Armstrong. Manjula is a senior regulatory consultant in the Global Regulatory Management team. She is an expert on regulatory requirements for quality control of biopharmaceuticals, including cell and gene therapies. She contributes to industry working groups, developing best practice guides and white papers on varied topics. So we are thrilled to have Manjula here with us. We also have Dr. Armstrong, who is the Global Head of Scientific and Regulatory Consultancy. She and her team are responsible for scientific and regulatory advice, fully supporting clients working with our BioAlliance testing services. Alison has authored several articles on trends in biosafety testing and is a member of regulatory task force groups related to rapid technologies. We are thrilled to have them here. Before I turn things over, and as you mentioned, we will be hosting a Q&A session at the end. So definitely submit your questions throughout the event. And if we don't get to every question, we will certainly address them offline as well. So that is it from my side. It is my pleasure to turn things over to you, Manjula. Thank you.
Thank you, Mackenzie, for that kind introduction. Good morning, everybody. So as the topic suggests, today we are going to talk about some of the regulatory complexities along the cell and gene therapy life cycle. Oh, sorry. Slides are not advancing. Okay, let's try this. Okay, sorry. Works now. Okay, so on agenda today are the manufacturing considerations for cell and gene therapies, regulatory intelligence in terms of what has been up and coming in the last couple of years, viral safety strategies, followed by product quality, and finally a summary of it all. So manufacturing. So before we delve deep into manufacturing of cell and gene therapies, which have been on the rise in the last decade, let's take a look into the two ways in which gene therapies can be realized. In vivo is where the therapeutic effect is by modifying or correcting abnormal genes by delivering specific DNA or RNA to correct them. The most common delivery system is based on viral vectors, which can deliver the gene of interest to the target tissue. So this is the in vivo. The other approach is to create cells which deliver the therapeutic effect. These can be autologous or allogeneic. Autologous using the patient's own cells, or allogeneic, which uses cells from a healthy donor. Here, the genes are introduced into the cells ex vivo, and then the cells are administered to the patient. Now moving on, so there are many key aspects to successful product development for such products, the cell and gene therapy products. The first is to demonstrate that the product is safe, potent, pure, and efficacious, and stable. The second is building quality into the product throughout the process by performing appropriate testing and characterization. Last but not least, have adequate manufacturing control by implementing GMPs to ensure product consistency and maintain the quality. So when these types of therapies started taking off about 10 years ago, the manufacturing structure was immature and manual processes borrowed from biologics manufacturing were implemented. There was safety and regulatory uncertainty due to limited understanding of the vector immunogenicity and long-term safety. There was high cost and limited access. There were quite a few supply challenges. And it was a fragmented development ecosystem consisting of small biotech firms with limited resources. Turning the clock around or clock forward to about 10 years to where we are today, the problems or the challenges have evolved. So there are still scalability limitations, but closed and automated systems have been implemented. Cost and efficiency pressures have sought to improve cell density, vector yield, and process optimization. There is a regulatory adaptation with accelerated pathways, but that comes along some challenges with CMC readiness and comparability. There's better access and better patient access, and payment models are still being explored. So here I want to highlight some of the solutions that the industry has developed that has made manufacturing more efficient. There are regionalized manufacturing hubs, especially for autologous therapies, with decentralized sites near population centers to reduce logistics and cost. There's platform manufacturing using modular scalable platforms for vector production and cell processing. Advanced analytics and AI are being used for monitoring and modeling to improve the yield. In addition, there's been partnerships between developers, CDMOs, regulators, and payers to accelerate access. I want to spend a couple of minutes on platform manufacturing. So platform manufacturing is defined as a process that leverages established steps, components, and documentation to accelerate development and production of new therapies for different diseases by focusing on the unique aspects of each product. So the key aspects include modularity, where a standard set of core components such as viral capsids, promoters, and manufacturing processes are used. Yes, shared infrastructure by the reuse of established manufacturing technologies, equipment, and raw materials. Standardized documentation. So pre-existing templates for regulatory dossiers and analytical methods. And the key is to focus on the differences. So isolate and efficiently develop the disease-specific components. The benefits of platform methods or technologies include reduced cost, reduced risk because we're leveraging prior knowledge, and speed to clinic. So now we move on to talk about some of the key regulatory requirements and some of the newer guidances which have been really targeted towards cell and gene therapies which target rare diseases. So As you start to move into the clinic with the therapies, one of the common questions is regarding what to do in terms of chemistry, manufacturing, and controls in each phase, and when to start implementing GMPs. In general, phase-appropriate GMP is accepted or expected. As you move through phase 2 and 3, process development and clinical study progresses, and so does the expectation for increased control on the manufacturing process. Assay qualification might be sufficient for the early phases, but by phase 2 and 3, or the pivotal phases, full validation of assay methods are required. And here we list some of the key regulatory guidance from the FDA on this topic. The EMA has a slightly different approach, although the basic principles are the same. The EMA published a Q&A guide to clarify their expectations regarding the application of GMP principles versus GMP or full GMP. The difference is that when GMP principles are expected, there is no inspection or certification of the facility, and it is up to the sponsor to apply GMPs. When GMP is required, the EMA will inspect and certify the facility. So here are two examples relevant to today's focus. For in vivo gene therapy, plasmid manufacturing can be done in using GMP principles. So wherever there is the dark green, it is the full GMP. The light green is GMP principles. So the cell bank manufacturing or virus seed manufacturing onwards, it is full GMP for in vivo gene therapy. For ex vivo, the establishment of cell banks for plasmid and or vector expansion, viral seeds, and the plasmid manufacturing and the vector manufacturing can be done by using GMP principles. The manufacturing of the genetically modified cell onwards, it is full GMP. Okay, so globally, now there is acknowledgement that CGTs are quite different from small molecules or mAbs. So there is now CGT-specific guidance, not only for like raw materials, but also for clinical trials and the pathways, etc. And I'll highlight some of these in the next couple of slides. So the US FDA published some guidance last year to further provide some guidance on animal-derived materials and also some FAQs for developing cellular and gene therapy products. This year, they have published some draft guidance, one on innovative designs, clinical trial designs for CGT products in small populations, and also the expedited programs for regenerative medicine therapies for serious conditions has been updated. In Europe, this year, there's the Formura 3186. It's a monograph on gene therapy medicinal products, and also another chapter, 5.34, on additional information of gene therapy for gene therapy medicinal products. So these provide good guidance on the quality requirements for these therapies. In addition, the EMA also published a guide on the quality clinical and non-clinical requirements for investigational ATMPs. In Japan, the Sakigake designation, or this is their expedited pathway, is applicable to cell and gene therapies. And as of May 2024, Japan allows exemptions from domestic release testing for imported regenerative medicine products if certain conditions are met. And now staying on the topic of product release and batch release testing, in Europe, the requirements include that the medicinal products must be manufactured in accordance with EU GMP. And a qualified person has to confirm that the batch complies with GMP, the product matches the specifications in the marketing authorization, and all required testing has been completed. For products that are manufactured outside the EU, they must undergo retesting in an EU-based GMP-certified lab unless it's covered by a mutual recognition agreement. And there are documentation requirements, such as batch records, certificates of analysis, which have to be reviewed by a QP, and then each batch is released for marketing. So you can imagine that when there are cell therapies or gene therapies that are imported into the EU, this requirement for retesting can become a huge challenge. And please keep in mind that no mutual recognition agreements are allowed for these types of therapies. So the EMA has recognized this fact or this challenge and does provide an exemption. So this is only allowed when there's limited amount of material available or short shelf life, and the testing in the third country should be conducted in a GMP certified facility. So if those conditions are met, then an exemption could be requested. This mainly works for autologous therapies or patient-specific therapies, and prior consultation earlier in development is recommended by the authorities. Okay, so now, before moving on to the next section where I'll hand off to Alison Armstrong, there'll be a brief poll that will be run.
Okay, hopefully you can see the poll. So we have question, what poses the greatest regulatory risk in your CGT program? Got a few answers coming in. I'll give it a minute. We've got about 70% participated. A few more coming in. So Alison, it looks like post-change comparability across sites and batches is the glaring answer here. Okay, we'll go ahead and close the poll. And I'm going to share the results. Here we go. All right, go ahead, Alison.
Okay, thank you for your participation in the poll. That's very interesting and really exciting information for us in terms of the kind of oversight, certainly around Manjula's summary and oversight. I'm going to move to a little bit different kind of approach in terms of our kind of navigating the regulatory kind of landscape. This particular slide really gives us a the nice review that Manjula has provided on evolving the regulatory landscape, the manufacturing and raw materials challenges that we see. But let's maybe just focus a little bit more on some specific topics. This is a very large topic to cover in a very short webinar. So certainly, hopefully, we have challenged and picked some topics that are suitable and relevant for industry. We absolutely have regulatory expectations, and they are evolving and continuing to evolve for the cell and gene therapies. And certainly we are seeing a large amount of publications and guidances around coronavirus safety and purity assessment, driven primarily by the ICH Q5A. And that goes from the testing right through to the viral clearance processes. And also another area that also is getting a lot of activity at the moment is really the potency topic. So again, there's some new draft FDA guidance for that particular topic, and there's an updated paper and regulatory landscape from the European authorities in terms of those approaches. Quality control continues to be a challenge. We need to understand the quality attributes, and we also need to make sure that we keep abreast of any technological awareness. So new technologies and alternate technologies that might be appropriate to address specific questions in terms of the safety and efficacy of the products. And of course, we are continuing to review and evaluate where there may be regulatory gaps or technical challenges, and looking at how these may be addressed, and we'll touch on some of this in the next part of the talk. We're looking at really the replication competent virus landscape, and also the fact that there's a very limited volume of material for many of these types of products, and that in itself can be challenging in terms of the application of test method. So we move to the next slide. This really summarizes to a certain extent the topic of this section of the webinar, which is around viral risk mitigation. We've probably seen that little triangle on the left-hand side of the slide in the past. So the prevent, detect, remove. But the table that is on the right of the slide gives you a little bit of an oversight of the different parameters and steps in kind of manufacturing process that are probably very specific to cell and gene therapies. So looking at the expression systems, raw materials, right through to the purification process. And there are various aspects that we apply and can apply in terms of mitigating and minimizing viral safety. Ultimately, what we have is we have requirements to prevent the introduction of contaminants within the manufacturing process, and that might focus very heavily on the raw materials and media preparation. We also want to ensure that as we are producing the materials, that we keep control of the potential of contaminant introduction. And again, that is through the detection through perhaps cell culture and or the raw materials that are used. Again, testing, moving a little bit upstream in terms of some of these products. And then the removal process is very much perhaps limited to certain molecules, but again, we can apply the specific downstream processes that are applied for this particular program. When we look at the next slide, we're really starting to look at some of the other safety considerations that are introduced. In particular, I've highlighted this as purity, but probably call this safety, if you will. So again, what we are looking at is the various different methodologies that are used throughout the production process. So ultimately, sterility is almost the cornerstone of many of the tests that we apply to make sure that we have products that may be used in patient population. And we're very fortunate that some of the alternate and updated regulatory guidances have been updated in line with the fact that we have modified materials, low volume materials, and tests that you require to provide test results in a much more rapid manner. And I've just highlighted here an example of a specific method that may be applied. And this very much has been introduced in industry after the update to the 21 CFR 61012 regulation, where really it gave more of an understanding of the changing landscape of such products, and where we actually have a level of flexibility in terms of choice of technology for specific to test and evaluate specific tests in the sterility environment. This has been updated recently by a new USP regulation, as well as updated regulation in the European Pharmacopoeia. Also, mycoplasma still comes to mind in terms of some of the types of contaminants that might be of concern, particularly for these particular product types. And again, using alternate and updated methodologies can be applied, in particular using quantitative PCR. And there has just been an update to the actual specific chapters within the European Pharmacopoeia, which has been undergoing revision for some time. Again, it allows us to use shorter turnaround times and sample requirements that are much more limited in terms of volume. When we look at the adventitious agent testing, Again, there is a requirement to provide that level of assessment and assurance around virus detection, but we can use different methods. And in particular, the next generation sequencing methodology has really again come to the fore in terms of application in this particular environment. We're able to circumvent maybe toxicity and neutralization issues by using that technology. And combined with an identification test can give you a lot of valuable information on the product in question. And last but not least, probably a topic which is probably very highlighted in an industry is replication competent virus testing. And there's been a number of different regulations and oversights just that we apply for that particular program. Perhaps if we move to the next slide, we can go into this in a little bit more depth. So the FDA published a guidance for industry document in 2020, and the European Pharmacopoeia have just updated and introduced a chapter 5.31. Both of these provide understanding of the test points for replication competent virus detection for gene therapy or cell and gene therapy products. That would include looking at your stable producer cells, your vector-containing supernatant, and perhaps, looking at purified harvest or final lot. The requirement is to use a permissive cell line for amplification and a suitable endpoint to really look at the high level of sensitivity that is expected for those tests. And again, the test methods that are applied here and the endpoint analyses tend to be relatively similar depending on which regulation you read. We're also pleased to see that in China that the CDE produced a question and answer paper which addressed some of the challenging questions that industry has on applying both the volumes of material that are required for test as well as the endpoint analysis for both replication competent retrovirus and replication competent lentivirus assessment. Some of the regional differences, the European Pharmacopoeia, Chapter 5.34, does not require testing on transduced patient cells. And probably just a comment is that the FDA guidelines are very focused on replication competent lentivirus and retrovirus and provide a lot more detail in terms of test points, the method expectations, and also the appropriate test volumes. We move to the next slide. We really are now starting to look at some of the requirements and requirements for identification testing. And that can be used to confirm the identity of a viral vector. It might be used to identify transgenes or even confirm patient cell identification. And this really just provides you a list of the types of assays that are being applied and used, both from a genotypic point of view, perhaps PCR or sequencing methodologies, or a phenotypic approach where we may use ELISA testing, HPLC, or even look at surface markers using FACS analysis. If we move to the next slide, we now start to really look at some of the introductory kind of methodologies that have been more recently applied, primarily in the space of the downstream process. So we're looking at the manufacturing programs. What we're now looking at is whereabouts could we basically, provide a level of risk mitigation in terms of viral assessment for some of the viral and gene therapies. So in 2020, there was a CMC document on human gene therapy, and that references robust viral clearance studies may be necessary to remove and inactivate adventitious agents. With the advent of the ICH Q5A revision 2 publication in 2023, this expanded the scope of that particular document and now introduced the ability to evaluate viral risk mitigation for, in particular, gene therapy applications. Essentially, this document again highlighted that the risk of contamination of adventitious viruses and residues of viruses during production with either by helper virus or protein expression systems may be mitigated using the principles and information that was in the guideline where it was at all possible. They also further highlighted that viral clearance validation should include model viruses representative of adventitious and endogenous viruses, and if possible, the relevant helper virus, and choose the appropriate virus models suitable for the production system in question. And I've just highlighted really obviously the considerations that we need to make for certainly non-enveloped viral therapies. Inactivation steps may be implemented, low pH or solvent detergent steps might be appropriate, and a large pore viral filtration may be implemented, particularly for AAV molecules. The lipid envelope viruses and cell therapies are somewhat more challenging. Your viral inactivation process would destroy your lipid envelope, and viral filtration is not really a viable clearance option. So there's limited options really for some of those more lipid enveloped viral and cell gene therapies. We go to the next slide. It just summarizes really the point around the non-enveloped viral therapies. Looking at an AAV purification process, we can provide viral clearance. Looking at various steps, so essentially we could effectively inactivate enveloped viruses through the detergent lysis steps by low pH. Basically, we can also look at the chromatographic steps and the large pore virus filtration, where we actually show that there is good ability to remove both enveloped and non-enveloped virus. We moved on to the next section, really just around some of the product quality assessments. Effectively, we're now really moving into more of the process and product evaluation programs. So we're looking at, for the next slide, the characterization of and some of the questions that are really asked around product and process quality. So how does my vector function? What is the physical characteristics and attributes of my vector? And what are the key points in terms of biological activity? Do my transduced cells, are they efficient? Does my transcription levels meet the requirements that I need? And is my protein expressed functionally? And is it active? So the next few slides just give us some ability to look at the different types of questions that are addressed and some of the technologies and methodologies that can be applied. So the next slide provides us with a similar level of evaluation for residue materials, so looking at the product quality and purities, perhaps looking at the evaluation of empty full capsids, nuclease, resistant host cell DNA, looking at resistant helper DNA, again, looking at the replication competent virus testing, non-infectious particles or aggregation. And you've got a list here of different types of assays that can be applied, fully validated, quality approved methodologies that can really address perhaps the challenging questions that are asked around the introduction of some of these particular impurities. If we look at the next set of slides, we again look at the evaluation of purities just in terms of very specific the residuals. So host cell protein, host cell DNA from your starting materials, residual cell culture related components, residual process reagents, benzonase, or some of the chromatographic ligands, and residual plasmid DNA. So again, the nature of the lot release test can really be provided in terms of specific approaches that can be used based on the application and tests that are applied, whether that be a host cell protein analysis, using immunoassays to challenge and look at the specific specifications that are applied for host cell protein and also for DNA, and various other types of methods that are applied for these particular product types. If we move on to the next step, really we're focusing a little bit more on the vectors. So a combination of methods are really required to be used, in particular to determine both particle number and infectious titer. And effectively, we'll look at genomic copies. We can measure those using looking at the viral genomes, using PCR-based methods. And if we're looking at the number of particles that are present, again, we can look at ELISA-based methods for that particular application. We can also look at the concentration of viral particles that can transduce cells, and that's maybe evolving infectivity assays, really appropriate for the cell substrate. And also, if we're looking at the virus measurement, we may be using different analysis points, flow cytometry, plaque, foci formation, or even PCR where applicable. The next slide goes into some of the potency requirements in terms of the application. So recently, as I've mentioned on the first slide, the FDA have now got a draft document that was originally introduced in 2011, has now been updated in 2023. We know that the modes of action for some of these molecules, cell and gene therapy molecules, present high challenges for the potency determination. Essentially, there are some significant changes to this document in terms of the themes between the original document and the new document. It looks more in terms of the concept of the overall control strategies rather than focusing only on the potency assays. And a matrix approach perhaps is less emphasized in this new document, where really we're looking at the use of one assay covering multiple steps, and looking at how this can be applied moving forward. This has been a considerable topic of interest from industry in terms of different industry forums, and also looking at the different the high difficulties of actually performing potency determination, primarily due to low batch numbers, sample volumes, the complexity of just demonstrating that biological activity, and then again, the feasibility of keeping to the concepts of the guidance documents brings into this a level of complexity. Again, it's almost a topic on its own. The next slide provides a little bit, I believe, around stability testing. And stability really around the updated ICH-Q1 update. It's still in draft, but it does acknowledge the difficulties that are expressed by the stability testing of ATMPs or cell and gene therapies, mainly because of the limited numbers of quantity of material that is produced. The scope includes really the standard types of products that we look at, which would be both autologous and allogeneic cells, gene therapies, oncolytic products, and it covers from drug substance, intermediates, and drug products. And it also emphasizes, again, that level of understanding of the critical quality attributes, both process and product knowledge. And it provides some examples of product type specific attributes. So for cell therapies, it's looking at viability and concentration. And for viral vectors, perhaps, as we've just mentioned, some of the methods I've just applied: particle number, genomic and infectious titer, potency. So really, the regulators are providing us a lot of information for these products, and the applications and the test systems that are applied and the oversight of the experiences that we are gaining as we move forward are really standing us in good stead. I think the next slide provides us with a summary of the actual overall presentation. So we have covered the manufacturing efficiencies, looking at different platform methods and advanced analytics that can be applied. There are global cell and gene therapy regulatory pathways, and these are being implemented. There are many documents that have been introduced to our industry and presented to our industry from the regulators, with great input from industry partners. This is really increasing apace on an ongoing basis. Viral safety strategies continue to be implemented. Based on the nature of the product is where you would apply those strategies, and alternate and updated technologies are very much to the fore. And the demonstration of purity, potency, and control of residuals and stability continues to be a regulatory expectation. So thank you for your attention, and we're very happy to take questions.
Perfect. Thank you so much, Alison and Manjula, for those wonderful presentations. So we'll have everybody back on for the Q&A here. Let's see, we did get some from the audience, so I think we'll start with those. And certainly your insights here would be appreciated if you have them. So Manjula, I think this one might be more for you, but Alison, feel free to chime in. Have we seen any changes or expect any changes to the guidance as a result of the changes to the FDA in the current administration?
So the current administration is the one who published the 2 drafts on the clinical trials and the expedited pathways. So they do have a heavy focus on rare diseases So cell and gene therapies definitely fall under the rare disease realm. So I don't think the seminal ones about the safety, purity, etc., they are going to change, but it's more like expediting the development of these. That's what they're focused on. So that really showed like through the clinical trials. And there's one more they're talking about. It's called plausible clause mechanism, or plausible mechanism clause, right? So you don't really have to have a fully understood mechanism of action, but as long as you have like a plausible mechanism that might, plus of course data to show that it's efficacious, then that might be sufficient to get approval. So those are the things that this new administration is talking about.
Perfect. Thank you.
Manjula, just to comment, I mean, I was very heartened when the new FDA regulator came into, director came into play. There was a really nice meeting of the kind of industry leaders and the FDA in particular, and that was really nice to see. We were actually able to have really nice, good, open conversations with the administration to highlight their challenges, their concerns. Yes. And it's nice to see that there are some movements in terms of those aspects. So
yeah, the listening sessions, right? Yeah,
correct.
Those
correct.
are really good. Yeah.
Wonderful, thank you. The next two questions are more, I think, around the MRAs that may not be in place. And the first one is with regards to the EU testing requirement that you had been previously talking about, Manjula. Does that mean that EU GMP material should be tested in an EU certified facility?
EU GMP material, is that what you're saying?
Oh, sorry, EU GMP should be tested. So I think we're specifically saying, if you're releasing in the EU, it means it has to be tested in an EU certified facility.
Yes. So for it's mainly it's for commercial molecules, right? When the products are in clinical trials, they're undergoing clinical trials, that requirement is not there. But if it is a commercial molecule and it's being manufactured outside the EU, then to be released for EU, public, it has to be retested and released by a QP who is based in an EU-based lab in an EU country, and then it has to be also GMP lab. So definitely that is required, except like I talked about the exemption. So if you can get an exemption based on the nature of your product, then you don't have to do it.
Okay. Perfect, thank you. And I think along those same lines, even though we do not have any MRAs between the FDA and EMA, are you seeing any collaboration between those two agencies?
There are definitely collaborations. There's also talk about a parallel review for some of these types of therapies, just because of the challenges associated with, small volumes and also just having that patient waiting. So yes, so there are like those parallel reviews that are feasible between, say, EMA and FDA.
Okay, good to hear. That would be valuable. Alison, I think this question is more for you. And it's, are there any specific purity tests focused on prion contamination?
Oh, gosh. So that's actually been a topic of interest for a very long time. There are methods that can be applied to detect prions. I think the unfortunate problem is they're not sensitive enough to pick up prion contaminant in, say, a raw material, because of the background materials. So there are methods that could be applied, but unfortunately they are not applicable. So we still use the consideration of the country of origin the geographical evaluation, a risk assessment that is used for that particular application. I mean, there were many studies performed for various different plasma products in the past, and we're using Western blot to look at that in terms of clearance potential. It would be quite an extensive piece of work to do that kind of work for these types of molecules. So I think people are looking for an off-the-shelf method. The methodologies have got much improved, but I do not believe they are still at the level where we're able to use them just as a standard test, a yes/no answer. We still have to use these risk assessment evaluations for prion evaluation.
Interesting. That was a new question for me that I hadn't seen. So thank you. Alison, I think we'll stick with you for this next question. For the approval of innovative technologies for cell and gene therapies, what data is expected by regulators to enhance the acceptance of alternate methods during filing?
I think, I mean, as with all changes or updates, the regulators want to see the information. They want to see the data. They want to see the understanding of the method. They want to be informed of how the method would run, what the methodology development perhaps has been informed. They will absolutely want to see validation data on the test system, and they'll also primarily probably see either equivalency or comparability testing where it makes sense. Many of the new technologies versus culture systems, it'd be quite difficult to do a full comparability, but certainly an equivalency test would be applicable. Probably conversation with the regulators in advance of making a change is always advised. I think that's probably one of the key topics that we hear a lot. And certainly where technology perhaps has been introduced to the regulators through some of their innovation pathways. So there are pathways in both Europe and US. So US have an ETT, an Emerging Technology program, and Europe has an innovation task force team that actually can meet. You can present the technologies or have that technology as part of their programs, and say if the method is then in your filing, to mention that it has been undergone that review. So there's a dossier presented and the regulators review that. That's a nice way of actually providing that update. I think, again, all I keep saying is it's data, data, data. They need a lot of information, and they want that level of confidence. And the more information, updated technologies, application can provide to vendors, customers, the better.
Mm-hmm. Yeah, absolutely. Thank you. Manjula, I think we'll jump back to you for this one. Is there any regulatory guidance on the use of platform processes?
Yes. So from the FDA, there is a guidance. I think it's still draft on so there's a designation. So there's a platform designation that you can get. So there is guidance on that. So what basically what you can do is, if a particular company has an approved therapy, then say the vectors and some of the manufacturing process that has been used for that, the approved product can be applied to other products, and then you can leverage the information around that to then make, you can leverage that, and then the approval would be faster because you are getting that platform designation. So, and I think there has actually been an example of a company who has been successful at doing that. At the EMA level, they are still working on it. They have, I think they've published a reflection paper, but they don't have guidance yet. I think it's on their list of things that they're working on.
Thank you. Let's see, Alison, we'll come back to you for another question here. We know potency is a hot topic. So what are the key topics under consideration when choosing systems to evaluate the mode of action addressed by potency test methods for these more complex therapies?
Yeah, I think I mentioned when we were talking through the webinar that the biological activity is obviously a key quality attribute that is required for these modalities, and tends to be measured by quantitative kind of biological assays that are capable of detecting really the mode of action over a concentration range. The methodologies need to be consistent. They need to be showing a level of accuracy and performance. They need to be sensitive to pick up small differences in biological activity, and they have to be very robust, which can be quite a challenge in terms of the processing. They also need to have to be relatively easy to perform. We're performing these on an ongoing basis. So once they're developed, they need to be able to be achieved within the laboratory systems that are available, and have the relevant controls and perhaps data, analysis that's necessary. And it might be more than one test that is necessary or required. Also, because a lot of these molecules are short shelf life, there might be a limited time for full assessment. There's definitely going to be limited volume. And also in quite a number of cases where we would maybe have references would be applicable and available for some of our more classic molecules, it's a little bit more difficult for cell and gene therapy products. So I think, again, it's just probably the recommendation, and we always explain this, is there is some complexity. It is achievable. You can do that, but think very early in the development of the molecule what types of potency questions you might need to address so that we can continue to provide support for the molecules as we move forward. It's a key assay that is required and methodology. So yeah. So hopefully that covered some of the thoughts, or my thoughts, on the potency test.
I think so. Like you said, this is a lot of information to fit into a very short webinar. So if there are more specific questions, or, you want to talk directly about your, therapeutic or application with us, like that's certainly encouraged after this event. We look forward to folks reaching out to us about those kinds of things. I think we'll take this one last question and then wrap up, being cognizant of our time here. Manjula, I do believe you touched on this with our previous question, but do the batch testing requirements in the EU apply to all clinical phases?
So yes, so the batch testing in terms of that, so batch has to be tested and certified by QP, but the requirement about the imported materials having to be retested, that is applicable only to commercial products. So the retesting part, if for imported molecules, that is applicable to commercial products only. Perfect.
Okay, thank you for clarifying again on that. I appreciate that. So, I think that is going to be it from our side. I will let Anne close us out here. So thank you again, Manjula and Alison. Really appreciate the time that you guys have taken for this valuable information. And Anne, I'll turn it back over to you.
Great. Thank you, Mackenzie. And thank you, Alison and Manjula. This was fantastic. A lot of great questions. So I'm really pleased with the outcome for today. And just to remind, whoops, my slide didn't advance, sorry. We will have another one in January. I don't have the date yet, but we will keep you alerted in our CASSS communications. And you will receive a short survey after this event, as well as the slides, and the recording will be available within a few weeks of the event today. So stay tuned for that information. Again, thank you again to MilliporeSigma, Mackenzie, Alison, and Manjula. It was a terrific presentation today.
Thank you.
Thank you.
Thank you.
All right. Thank you, everyone.